Now that we have discussed the structure of the neuron, let's add a note on the basic function of a neuron. So a neuron is capable of receiving signals. Now once it receives signals, it analyzes these signals. It decides which information is important and which needs to be filtered out. So it integrates and processes information. And then it transmits this information to target cells, which could be other neurons, muscles, or glands. So by performing all these functions, it serves our broader level of function. The highest ability of the human being is our ability to learn and to remember. So these information processing and learning abilities are served by the neuron. Let us now discuss the important properties of nerve fibers. So the properties that we discuss in this section are excitability, conductivity, all or none law, refractory period, summation, and accommodation. Let's now discuss the first property, that is excitability. So what do we mean by excitability? Now this is a property that is unique to living cells. So excitability is the ability of a cell to respond to a stimulus. It is what differentiates a living cell from a non-living object. So in that measure, a neuron is highly excitable. Now, how do we measure excitability? Think of it this way. There are some people who are highly excitable, which means that they need very little to become excited. Now, the same way, a cell sometimes needs very little current to respond. So then we call it as a highly excitable cell. So in this context, we define a term called rheobase, which is the minimum strength of current required to elicit a response. It is also called threshold stimulus. But there is another factor which is equally important, and that is the duration for which you will apply the rheobase. So the minimum duration for which the rheobase is applied to elicit a response is called utilization time. Now this means that if the rheobase and the utilization time are low, then the cell is highly excitable because it is easily responding to the stimulus. Now in the laboratory setting, it is usually not very practical to apply rheobase because this is too small a current. So we generally apply a current whose strength is double the rheobase. Therefore, we define yet another term called chronaxy, which is also a measure of time. So chronaxy is defined as the minimum duration of time for which twice rheobase current must be applied in order to elicit a response. This is what we usually use, the chronaxy, as a measure of excitability. Now, this is how we plot these parameters on a graph, and this graph is called the strength duration curve. So as you can see on the y-axis, we plot the strength of current, and on the x-axis, we plot the time. So initially, as you increase the strength of current, there is no response in the tissue. At this point, the tissue starts responding. So this is the rheobase. Now, from this point, if you drop a vertical, that will give you the utilization time. This entire period is the utilization time. Now, to plot chronaxy on the y-axis, you mark double the rheobase. Draw a horizontal to intersect the graph and then drop a vertical. So this becomes the chronaxy. So on this graph, you have now plotted all the parameters that we talked about. So this is the strength duration curve. Now the excitability of a cell can vary depending on different factors. For example, on a hot and humid day, we often experience muscle twitches or nerve twitches. So this is related to electrolyte changes, temperature, etc. So the most important factors which affect excitability are temperature, pressure, pH, chemicals, concentration of ions, and the blood supply to the tissue. Now this slide gives you the Chronaxi values of different tissues. So you can appreciate how different tissues differ in their excitability. You can see that when it comes to the large myelinated nerve fiber, its Chronaxi value is the least, 0.1 milliseconds, as opposed to the cardiac muscle, which has a Chronaxi value of 1 to 3 milliseconds. So the large myelinated nerve fiber is much more excitable than the cardiac muscle fiber. Now we talk about the second property of a nerve fiber, which is the all or none law. So when an excitable tissue is stimulated, there is either generation of action potential of maximum amplitude or no generation of action potential at all, depending on the strength of the stimulus. This is the all or none law. So let's examine this law in more detail. What does it mean? If you apply a sub-threshold stimulus, a stimulus which is of very low strength, you do not get an action potential. But if you apply a threshold stimulus, that is when the tissue responds with an action potential. So you get an action potential of maximum amplitude. Now you increase the strength of the stimulus further and apply a supra-threshold stimulus, you still get an action potential of the same amplitude as before, which is already maximum. So this means that you either have no action potential or you have action potential of maximum amplitude. There is nothing in between. So this is what the all or none law means. Now this is a graphical representation of this law. So you can see that when you apply a sub-threshold stimulus, there is no action potential. You apply a threshold stimulus, you have an action potential of maximum amplitude. Now, if you increase the strength of the stimulus, you still have an action potential of the same amplitude. Now, you must remember that the all or none law is applicable only to the single nerve fiber and not to the entire nerve bundle. A nerve bundle is made up of many nerve fibers, and each nerve fiber has a different threshold for the stimulus. So the all or none law cannot be applied to the entire bundle because the thresholds are different for individual nerve fibers. So remember that only a single nerve fiber obeys all or none law.